My name is Wilfred Abincha. I'm going to take you through this tutorial. Kindly subscribe, like, and share. Today we are going to look at structure of DNA. DNA stands for deoxyribonucleic acid. And the DNA is very essential, it's important to the life of organisms, and therefore we have to study it. DNA is a molecule that carries genetic instructions, and these instructions are involved in development, functioning, growth, and development of all organisms. The structure of DNA, when you look at it, it's composed of two chains. This one here and this one here. You can see it's made of two chains that are coiled to form a double helix. So when these chains are coiled, they form a double helix. This is a double helix you are seeing here. So each of these chains is made up of polynucleotides. It means a lot of nucleotides are joined together to form a polynucleotide. The DNA has a backbone, and this backbone is in this chain here. This is a backbone here. So this backbone is made of sugar and phosphate group. This sugar is a ribose sugar. This is the structure of a ribose sugar. It has 5 carbons. This is carbon number 1 here, 2, 3, 4, 5. So this ribose sugar at carbon number 2, this carbon number 2 here, this oxygen is removed here. So that's why it's called deox, because this oxygen here at carbon number 2 is removed. So it's a deoxyribose sugar. So the backbone of DNA is made up of a deoxyribose sugar and a phosphate group. We have seen sugar, and now let's look at the phosphate group. And this diagram here, you can see this is a deoxyribose sugar here because we don't have oxygen in carbon number 2. And this carbon number 3, number 4, we have carbon number 5 here. In carbon number 5, a phosphate group is attached, this carbon number 5. So the backbone of this DNA is made up of sugar and a phosphate group. Phosphate is attached at carbon number 5. That's why you see in this structure we have written sugar and phosphate. This is a backbone. And in the sugar and the phosphate group, a nitrogenous base is added. When a nitrogenous base is added, it forms a nucleotide. This is a deoxyribose sugar because it doesn't have oxygen here in carbon 2. And this is a phosphate group added at carbon number 5. It's carbon number 1, number 2, number 3, number 4, number 5. At carbon number 1 here, a nitrogenous base is added. So this molecule here, which is having a ribose sugar, a phosphate group, and a nitrogenous base, is called a nucleotide. These nitrogenous bases are here. They can be divided into pyrimidines and purines. A purine has 2 rings. The form of purine, you can see these purines have 2 rings, guanine, adenine. They have 2 rings, while pyrimidines have only 1 ring, thiamine and cytosine. Ideally, a purine binds with a pyrimidine. It bonds with a pyrimidine, so to make a perfect bond. So these nucleotides, they are joined together. That process is called polymerization. When they are joined together, they make a polynucleotide. And a nucleotide joined with another one, makes a polynucleotide. If you can see this, a polynucleotide here, and here we have another polynucleotide. At carbon number 5, we have a phosphate group. Also here at carbon number 5, we have a phosphate group, and here we have, this is a pyrimidine and this is a purine. These are bases. So these are 2 nucleotides. So to make a polynucleotide, this nucleotide has to bond together, has to bind. This process is called polymerization. Here at carbon number 5, we have a phosphate group which joins with oxygen in the third carbon of the next nucleotide. Phosphate of one nucleotide joins with carbon number oxygen in carbon number 3 nucleotide of another nucleotide, and this oxygen points with this phosphate group here. Here remains hydroxyl and hydrogen atom here. This hydroxyl and hydrogen atom will bind together to form water, and this process leads to formation of this. This is a polynucleotide formed of two nucleotides, plus water is lost. So this bond which is formed between oxygen on carbon number 3 and phosphate which is on carbon number 5 is called phosphodiester bond. And this defines the direction of a DNA backbone. So it runs from the 5 to 3 direction. That means this 5 carbon joins with this 3, and another 5 one will join with the other one of 3, and this forms this gene here. This is a nucleotide here, and this one is another nucleotide, and this is another nucleotide, and this is another nucleotide, and these are bonded at from 5 to 3, 5 to 3, 5 to 3 direction, and this when they are bonded these through polymerization, they form a backbone of DNA. So you can see here we have this nucleotide and this one making this backbone, and from the other side we have another antiparallel backbone moving this direction. You see this is moving from 5 prime to 3 prime, and this is from 5 to 3 prime. You see we have bases here, a purine and a pyrimidine binding or making a bond here through hydrogen bonds here. As you can see, cytosine and guanine are having 3 bonds, and also you can see this here adenine and thymine, they are making here they are making 2 hydrogen bonds, and when this structure here coil in a way, it makes this double helix. So from this bond we can say that if your DNA you analyze in a way and realize that you have more of these points, guanine and cytosine, it means you'll need a lot of energy to separate the 2 strands. That's the end of this today. If you like this video and if you find it helpful, kindly subscribe.